and he is a young conservationist, completing a master's in biodiversity conservation in natural resource management, and she has gained a lot of trainings in entomology, where she is doing research on entomology, especially soil-eater insects. That's his focus mainly, and he's going to talk about a good and a very interesting topic, which is entitled Assessment of Arthropod Diversity and Community Composition in Forest Plantations, case study of Arboretum of Ruhande, Southern Rwanda. And I would like to highlight that this topic is his topic of research in his master's thesis. And now he's going to take us through this interesting topic and tell us what he found and how he did this research. You are welcome, David Buyoya, and I thank you for agreeing to speak to us. And we would like to hear from you. Over to you, David.
Thank you much, Vena, for such a great introduction, for what I'm doing, even what I have done in the past. I was going to first to thank for CoEB for giving this opportunity, and also for this journey of 3 years of internship here at CoEB. Allowing me to share I hope you are seeing my screen.
Yes.
Yes, I see it, David.
Okay, thank you. As Vena have said, I'm going to share my research for masters that I'm going to complete soon, where I was taking in the program of biodiversity conservation and natural resource management. This research I have been conducted through the supervision of Associate Professor Venuste, Director of CoEB, and also Dr. Uwizerimana Jean de Dieu, lecturer at the University of Rwanda. My research topic was to assess the arthropod diversity and composition within Arboretum of Ruhande forest. So a bit. Are you still seeing my screen? Okay, thank you. Firstly, most of people were asking me, why are you doing your research? What have been motivating me to do a research in Arboretum? Firstly, because when we are talking a forest, most people or some people are not aware that the forests are being provided many things. When we are talking about forest, some people are aware of the timbers or charcoals that are being collected within the forest, but also the forest have many function to us humans also, and also even other animals, such as we have a medicine we get from the forest, medicinal plants for those who know about ethnobotany, they know that the forest we can easily to get the medicine. And also when you look on ecosystem services that we are getting from the forest, like carbon sequestration, when we talk about soil erosion, avoiding of soil erosion, those are also main function that motivate me to take my research in a forest. We all know that we have many habitat in Rwanda. We have a wetland, we have agriculture, but for me, I'm motivated by looking on the importance of forests, and also the reason why I choose Arboretum. We have there some conditions that allow to have a good research compared to the other forests where it is easily to cut. Backing to the arthropods, even some people are not aware of those small insects. Some are, why can't you have a research on big animals? We have gorillas, we have another animals. But for me, as Vena have talked about, I spent almost 3 years at CoEB working in entomology. The motivation is that arthropods are among the diverse group. When we compared the animals we found in this taxon of arthropod, they are many. By looking on number of people who are working in entomology, we still have few peoples who are working on those insects. That's also the motivation for taking my research in these areas. Backing to the advantages or the role that those small insects, as you have seen on the below figure, we can look at beetles. By looking those small beetles, their role in a given habitat where they are working as decomposers for all those foreign leaves or also those all the plants that are being found within the forest. Their role is to decompose. Imagine if we don't have those animals within the forest, we may have many big trees that are being on the ground. So those animals are helping to decompose those organisms. And also, when we look on the soil, when we talk about those insects, they are helping us within soil aeration by decomposing those leaves, those plants, and also mix with the soils, and it make the structure for the soil. And also they are good indicators for the health of a given habitat. It is easy for we as a conservationist to look on a given kind of species and easily to assess what the health of a given habitat. But even though we have talking about those their laws, as we all know, every day there is increase of humans. As we human are being increased, we also need some areas to use. So as we need those areas, some forests are being transformed in agriculture, other areas that are being used by those animals are being used for urbanization. By looking on what we need from the forest, we need charcoals, we need timbers, or those human needs, human depending to the forest, we are being cutting those forests. As we change their habitat, so their population results to decline. Sorry. Let's move on another research by looking on another research that have been doing within Rwanda. We have found that most of research are being carried on native and exotic species, three species, by looking on the study done by Segimana in 2021 on native forests where they have been get the native forests to have been high diverse and even
Yeah, colleagues, bear with us. We are having an internet problem, but Buyoya is solving the problem very soon. So give us a minute and the problem will be solved.
this is almost
Yeah, I am here. He is back.
uh, sorry we face the issues of electricity. We have been using uh, but we solve it. So I was talking about the research that have been carried previously. We have found that the research done by Sengimana found that the native forest to have the diverse of arthropod compared to other plant of exotic. And also the research carried previously found that the sampling method that have been applied to collect those insects are also influencing the observed insect. Looking on our study area of Arboretum, we have different research that have been conducted within this area. We have the seasonal variation of little arthropod in some eucalyptus done by Sabimana, and also we have different research done by Venuste on the impact of forest and agricultural land use of soil and little arthropod, where they was compared their insect of Rwanda site and Arboretum. We have also different research card on different land use where they was comparing the arthropod of here Arboretum in forest and also different land use like banana and coffee plantation. And also currently research done in 2021, we have another research carried out by Buyoya, where he was looking on long-term effect of forest plantation species on chemical soil properties. And also recent study that carried out was look on three species diversity. From all above research carried out within Arboretum, here we have found some of gap to be addressed. Are you still seeing my screen?
Yes.
Okay. I'm facing the issues of moving to the next slide. So let me look how to solve it. I hope they are back. I was talking about from all those research have been carried out within Arboretum. We firstly saw that there is no research looking on combined introduced tree species, native tree species, even mixed tree species, because within Arboretum we have all those plots. The recent research that have been carried out, they have been conducted within native and exotic. For us, we was also encouraged to do a research also in mixed tree species. And also on looking the previous research that have been conducted within different areas, they didn't show us the effect or the impact of those sampling methodology that have been carried out to know how they are influence the observed abundance, also the species. So that's where we came up with the idea to continue with our research. Our research objective with design was to know their diversity and species composition of arthropod within those three categories of tree plots that are found within Arboretum, and also to assess the impact of tree species on the abundance and diversity of terrestrial arthropod. The third one, as we mentioned in our research gap, is to know the effect of sampling method on the observed arthropod. On this figure, we saw where we set our plots. As we look on this figure on the top right, we have different sampling points where we samples. Within Arboretum, we have choose to sample within 4 different plots of among those 3 categories. We have the red color that showed the plot we choose to sample for mixed species. We have the blue colors where it shows the plot within introduced 3 species, and also we have the green color that symbolizes the native plots. As you saw, we try to cover the whole arboretum where we chose the plots that cover at almost the arboretum. As we all know, arboretum have more than 524 plots, so we didn't manage to cover all, but we take some representative plots. By looking on those figures below, it shows 5 different subplots. Within each plot, we try to set 5 different subplots so as to cover each plot by taking different samples within each. The plot of Arboretum, each plot has the size of 50 meters squared. That means that this below figure, which symbolize 5 subplots, have the 50 meters. So as you look, we set each subsampling point by moving 5 meters of edges to avoid any bias from the insect you can collect from another nearest plots. Moving to the methods, we have tried to use almost 5 methods to cover each plot. We set many methods because we just want to know their influence on those arthropods we collected. On the first figure, we are getting the image of setting winkler. The winkler extraction, the way we collect those arthropods, we collect leaf litters with the soil which is mixed with upper layers. Then we sweep through the mesh wire sweeper so as to collect the top soils with litters with insects, but to remove those big leaves or big trees we can collect on it. And we place within those cost mesh. As we look at the white one, we use this one because we normally want to change their environment. As we collect those insects, we try to change their environment. That's why we collect with the soil. The second image shows the sweep. As we all seen, those insects that are on that dwell within the herbs, the shrubs, or within short or dense vegetation, we sweep within with sweep. So we collect those insects, we put them in the vials so we can identify in the laboratory. The next figure shows Japanese umbrella. Some people they call it beating net. So the way we shake the branch of the trees for the purpose of collecting arboreal species, those species that can be within the upper part of the trees. So we use those white sheet because some insects are too small in order to look at. We put the white one. The next figures is hand correction. This method, it targets those arthropods that are under the rocks or dead trees, or those insects that are moving within leaf litter layers. We collect it by our hands or forceps to collect it all. The down figures shows the pitfall. This method, we set it within the soil so as to capture or to target those insects that are moving during the night or day once we leave those plots. All those methods was being put within vials filled with ethanol. So we use ethanol for preservation purpose. The collected specimen was identified within laboratory of CoEB, where we use light microscope. As you saw, all those images have been captured using our phone. Those images was seen within the microscope. The purpose of microscope, even though they are insects we can collect within our hand, but we want to know they are really parts for easy to identify it. And also we use morphologically their features, how their legs, how their eyes are, their antennas, to differentiate those species. And also we identified to the family levels. We didn't reach to the species level. During our analysis, we use other software to analyze those collected data, and also we calculate richness, diversity, Shannon, Simpson, and also we try to use generalized linear model to know how they fit on those arthropods. Moving to the part of our result, by looking all collected specimens, we have collected 6,529 individuals, where was belonged to 21 orders, 88 families. By looking on those category of 3 species, the introduced 2 species, those plots we collected, I have shown those 4 different plots. The all total individuals we collected was covering 40.17%, where they have almost 70 different families. The indigenous was considering to have that 6.67 with 70 families, and also mixed species have the lowest individuals with 23.16%, and also 74 families. By looking on the below graph, of accumulation curve where it shows the family richness and total individuals collected within the arboretum. This curve, it shows how the effort our sampling was. By looking on the richness, the family we got, and also what can be if we continue sampling. So this extrapolation, the dashed line, shows that once we continue to correct how our result could be look like. And also as you shown, as you see, the mixed has the highest species richness, family richness of almost 74. By looking on the extrapolation, it shows that once we continue up to 5,000 individuals, it will also continue to get those species. By calculating the diversity indexes, as you see that the Shannon, the Simpson, even the evenness shows that the mixed tree species have the highest Shannon even once we look on richness. We have many family richness within mixed tree species. Moving to community composition, as we said, our objective one was to look on diversity and even to the community composition within those three different tree species. We found that almost the family we corrected was similar. Even this figure, that's what shows. It shows that the finding families was almost the same. That's why even on the significant was non-significant. That means that the possibility to find one family from another category of plot was high. That's how there is no difference within collected families. Backing to the relationship among arthropod abundance and family richness, by looking on correlation, the linear correlation, we found that the mixed tree species have the highest once we calculate the R from R squared. We found that within mixed tree species, there was higher correlated. By looking on all the linear line was upward. That means that the abundance and richness was positive. We call it negative once we have downward line. So our abundance and richness was correlated. As we sampled, we also have possibility to continue getting another families. Continue on looking the effect of collected arthropod on the plot and even methods by looking on how a given numbers or collected abundance are being correlated with the methodology used or on different plant. We found that within indigenous or native tree species plot we set, they have been highly significant. by looking on the abundance, hand collection, pitfall, and winkler was significant. But looking on collected abundance within Japanese and sweep was not significant as we look on their p-value was above 0.05, the one we set for significant. And also by looking within indigenous and mixed tree species, their value or their abundance on all collected methodology was not significant. Unfortunately, in indigenous trees, only three methods have been get their significant in the abundance. By looking on richness on family we collected, SweepNet has highest family richness compared to other methodology have been used. By looking on within introduced, we have on left table 49, 42, 42. By comparing to the hand collection within indigenous only 27 family richness. By looking on mixed, we have 20, 24, and also within introduced, we have 21. By looking on those values, we found that there is difference within methodology we have been used. Moving to discussion, within our collected data, we found that mixed tree species highest diversity of arthropods due to its wide range of niches. By combining mixed tree species and native tree species, it is easy to have different arthropods, insects that can be inhabited within those proteins. Firstly, looking on their food source, on looking their niches within this mixed tree species, we have many possibilities to have the many insects. Looking on the quality that are being, when we talk about on habitat quality, we mean those food, those niches, those types of tree that are being within those plots. They favors to have many arthropods compared to the mono trees of native or mono trees of introduced tree species. From our finding, we have found that within introduced species, we have lower arthropod by looking like Copressus funebrensis. When you look on their understory vegetation, we found that they are almost the soil. When we look on their ground, it is not easy to this plant to allow any vegetation or any herbs to grow on nearest area. This limit the number of insects that can be inhabited within this plant. And also when you look on indigenous tree species, as we have seen within our data, collected data, we found that it was almost within the middles. When you look on their abundance, when we look on their diversity, there was in the middle. That's why for indigenous, they allow to have many organic matters or biomass that are being within within those plot of indigenous. And also those organic matter allow to have many insects, but by looking on the what the insect get from the mixed, they have been high insect within those mixed species. Moving to the methods, sampling methods, by backing to both results. Once you look on the abundance, some methodology like Winkler and Pitfall, they have high collected arthropod abundance compared to hand collection and Japanese that have few abundance. But moving to the collected family richness, those methods like SweepNet and Japanese collect many richness or collect many arthropods. But moving to the abundance, they are the one that collect small or few numbers of arthropods. That means that when you're looking on the way those methods are varying, we come to conclude that no methods is no single method can be perfect to correct abundance and diversity. So we mixing the methods for correcting favors to get a good result, because some method cannot correct the high diversity, but for those who are looking on the abundance, other methods can be collected. Through this research, we recommend the identified species to continue to the genus. As for us, we identify only to the family levels. So next research can continue to the genus where possible to the family to the species levels. Moving to the, even though we was talking about vegetation, but also we recommend next research to look on also those soil content within the soil, other environmental parameters like pH, moisture, temperature. All those also can influence the observed arthropods. So the next research can continue, also can include on these environmental variables. And also we recommend next or other research to continue on looking on seasonal monitoring. For us, we have been collected our data during dry season, so it will be a good if we have both wet and season, but because of time for collecting data, we have been, it was almost short period to cover our thesis, so that's why we chose to use single season for our survey. I could end by thanking the CoEB and also University of Rwanda for the support for this research, for CoEB to give us a laboratory to use, and also to support for financially doing data collection. We also thanking RFA for allow us to do our research within Arboretum. Thank you very much for your kind attention.
Yeah, thank you very much. Thank you very much, David. This research is very interesting, and I'd like to suggest that when you are doing recommendations for this kind of research, try also to think broadly on what your data means for ecosystem restoration and other real life experience. Uh, because when I saw, for example, your first the results of on your first objectives, I thought about the projects like Combio and other projects who are doing restoration, and there is a big debate over there whether to plant native tree species, exotic, mix, if yes, at which percentage. Yeah, which means this kind of study tells us that we can better enhance biodiversity on working landscapes if we mix native and exotic plant species. And your study didn't tell us something about the proportion of native and exotic tree species, but at least we have got the idea that mixed trees can produce more biodiversity, at least for solitary arthropods. Yeah, that's my observation. And I would like to give the microphone to my colleagues here to also comment or ask questions. Thank you. You can either write in the chat room or raise your hand so I can give you the time to speak.
As we are waiting for comment and question, so what you was talking about, yeah, for this research, as we saw, the mixed tree species have the or allow us to have many insects. Even though we was talking about the arthropod, but also when looking on different animals that can be used. So for those projects, I can recommend to plant the mixed because it is easy way to get even financial by looking on social economics.
Thank you very much, David. Did you also try to see the proportion of indigenous versus native tree species in your sampling areas?
Sure, we have seen by looking on indigenous and also introduced, even though we found that the indigenous has the highest compared to introduced. By looking on these figures, when you look on Shannon for indigenous and introduced, we found that the indigenous has the high compared to the introduced, even though mixed tree species has the high. We look on also its proportion.
Not sure. Were you talking about were you talking about the proportion of native tree species over indigenous tree species, or still you were talking about solitary arthropods you sampled?
Uh,
That graph, sorry.
So we only look on arthropods.
Okay, yeah, I see. Yeah. Someone method? Yes. You're welcome, method.
Uh, Thanks, David, for a good presentation. I think can you hear me well?
Yes.
Yes.
So maybe first of all, I think there is a point where you say there is a significant difference, and then I mean you stress that there was a bigger difference between what and what, but yet the statistical test showed that the difference is not significant. Maybe to clarify whether you have a difference, a number of individuals, 500 and something and 400 and something, then in statistics, when it says that there is no significant difference, it simply means they are like similar. So that's the interpretation. You want
Yes.
to stress saying that they are significant while the statistical test has shown the opposite. Yeah, I think they are at that point. Then
Thank you. You can.
maybe so that we can go faster, you can answer the question as I ask, and then we move it to the next. First, I
Yes.
wanted to ask uh, when you were comparing the methods, their efficiency regarding the abundance or species, I mean family richness. So, how did you ensure that the methods are comparable? Because sometimes it depends on the time you invest on it. When you are using a sweep net and you compare with active active collection, how do you ensure first that they are comparable?
Thank you for a good question.
I mean, so that is sampling effort is the same for those methods.
As I have said, for each methods, they have the way to collect. Even though we spend SweepNet for 15 minutes, but for Winkler or or Pitfall, it takes long time. But as our methods have applied on all plots, we was sure that even though we have not taken the same time, even though we can't have the we can't apply all methodology on the same time. So because it is the way those methods are being applied, we prefer, even though they are not the same type, the effort was different, but we concluded to compare as it is the way data are being collected.
Yeah, sure, maybe there is some reference, but it's obviously it is more logical to compare those observations between sampling plots rather than between the methods. But possibilities also justified. So the
Yeah.
second, and it's very important, what's the motivation for identifying at the family level rather than a species? You know, in conservation, we aim to conserve the species. What are the constraints that we do not go further to the species level? And what's the plan next? Because possibly you have kept some specimens, and maybe you need to you can do further research. Why didn't you do go till species level?
Thank you for a good question. The reason we identified those species to the family level, firstly, it is because we don't have many experts for all those families. Once we have, for example, here in CoEB we have expert in ants, we have expert in dipteras like Randodier. So by doing our research to the family level, we have the aim of collecting many species, but for as this study going to continue, we have chosen some family so we can identify those collected species. I mean, family, those collected specimens, so we can continue to the species level.
So I recommend that you continue to the species level.
Sure.
So that possibly you can also promote some publication out of it.
Yes.
And remember, I will be there. They can assist.
Thank you.
Publication. So the last, what is so unique or a key contribution to the existing knowledge from your research? Something that you can say like a spotlight. I have found this special from my research, an observation.
From my research,
yes,
I can say that for other research that have been conducted, not only in Arboretum, there was only considered native tree and also exotic, their influence to the arthropod. But for this research, we also look on mixed tree species. So I think that is the unique from this research we conduct.
Thank you so much.
Welcome.
I think that's a contribution, David, and as you see, it's very interesting to consider also mixed forests, and particularly I can recommend that future studies can also do or sample in native trees, in exotic trees, and in mixed to see the difference. And in the future, for maybe you and the other colleagues here, if that's possible, it will be great to have the proportion of both, the proportion of native tree species in the mixed forest, because that can also inform the restoration activities. I can tell you out there, some people say we are going to plant 50% native, 50% exotic. Other people say we are going to plant 30% native tree species and 70% of exotic tree species, depending on different factors. But we can have like a good proportion when we can recommend to use when you want to promote biodiversity, because nowadays restoration is getting momentum and it is getting promoted, and the people want to promote biodiversity today differently from the past, because uh, in the past we were doing restoration as if we don't understand the term itself. Yeah, Method wants to come back. I want to hear from him, and also I will open to other people here to tell us their question or comments before I close. Method, over to you.
I forgot to put the hand down. Sorry.
Okay, no problem. Yeah, is there someone else who has a question or comment before I go on?
Hey, guys, you are so silent.
Yeah, they are silent,
You should
but
contribute.
let me hope that they were very interested in the topic and they understand everything. Yeah, Sarim, can you tell us your experience with Arcos? How much, how many trees? Tell us about the proportion of native tree species over exotic. What do you promote actually? From your experience, I'm not going to tell you to speak for Arcos, but I want to tell you, I want you to speak about your personal experience with Arcos and the other people maybe out there. I saw also the hand of someone, but
Edmond.
Edmond, yes, Edmond, you are welcome.
Thank you so much, Vena, and David, for a very wonderful presentation. Seriously, this is a very nice study, and I think your presentation was very excited to me. But I want a little bit to support you on what Method asked you about identifying those arthropod to species level. Honestly, it's very difficult to identify arthropod to the farm level because you can't be an expert in all arthropods. Maybe you can be an expert in Coleoptera, another person can be an expert in ants, but I think in this world we don't have any entomologist who can identify the solitary arthropods to the species level. So for the data that you have, if you want to go to the species level, it's better if you do DNA sequencing rather than morphological identification because almost they look similar. And you can think that is one species which is similar to this one, but when you do the DNA sequencing, you found that they are different. So it's what I wanted to add on. And thank you so much for your wonderful presentation, David.
Thank you, Edmond.
Yeah, thank you very much, Edmond. That's a nice contribution. I don't see any other hand, and I don't see any chat room message. Maybe I will close. I wanted also Salim to tell us about his experience, but maybe he's busy. Yeah, if there is no further question or comment, I would like to thank my colleague David for the nice presentation and the nice work actually. And I wish him to keep up the good work, continue the journey, dive deep, and maybe other studies will go to the species level as resources are being available and also time. Yeah. Thank you very much for attending the seminar today, colleagues, and thank you, David, again for speaking to us today.